Hi dear students, a warm good morning to all of you. Welcome to one more session of microscopy, and we are dealing with the different types of microscopes, and today we're going to talk about a new type of microscopy called the fluorescence microscopy. Now you might be wondering, what is this fluorescence microscopy? Now, if you look into this picture, you can see that the specimens which are being viewed under this microscope are being found to be fluorescing, and they are having different colors. Now, you can see green color in one, and some of them is being found to in another specimen which has been viewed. You can see red color as well as light green color also. So, and they are being fluorescing against a black background. Now, what is fluorescing? Fluorescing is emitting a particular wavelength of light and becoming colored. Okay, and not all particles have the ability to fluoresce. Now, if you go to look into the biological samples, you can see that various biological samples like chlorophyll, some oils and wax and all that, they will fluorescence. They have an autofluorescence. But not all biological samples have the property to fluorescence or to fluoresce on their own. So what do we do is, we coat them with some fluorescence emitting molecules called the fluorochromes. Now you might wonder, what is the property of fluorescence? When some substances are being exposed to a particular beam of light, they will absorb that light and they get what? Excited, and they will emit another frequency of light which will help us to detect them. I'll give you a simple example. You might have already noticed in the watches as well as in the stickers which we use uh, of the sun and moon and all that, which we stick on the walls for children to play. You can see that uh, during the daytime, these particles, the green color particles, they will absorb the visible light. And when you switch off the light in your room, or when it becomes night, the stickers of the star and the moon which you have stuck on top of your room will start fluorescing or emitting certain light. So this is a case of fluorescence where radon is absorbing the visible light during the daytime, and or not during the daytime, or when it was exposed to light, it was absorbing the light, and when the lights were switched off, it is emitting that light. So that is a case of fluorescence. Now in the case of fluorescence microscopy here, as I told you, not all biological samples have the property to fluoresce. So what you do, you coat your molecule, your specimen, with some dyes called the fluorochromes. Now what are these fluorochromes? Fluorochromes have different colors and they have different properties. Some of them bind to DNA, some of them bind to the surface cellular components. Like that, depending upon what you want to view, you can use different fluorochromes. Now, acridine orange and diamidino 2-phenyl indole, or the DAPI. Okay, these are ones which will bind to DNA or stain the DNA. And FITC, or the fluorescent isothiocyanate, it often attaches to the DNA probes or to the antibodies that are specific to the cellular components. So you can, if you want to identify a particular cellular component, you can attach an antibody which has been attached specific to that cellular component, and FITC has a property that it will go and bind to the antibody specific to the cellular component. And then also you have TRITC, or rhodamin, called the tetramethyl rhodamin isothiocyanate. It attaches to the antibodies that bind to specific cellular components. So depending upon what type of fluorochrome you are using and what is the purpose of your study, you will get different types of fluorescence as we had observed earlier. Now let's move on. So, in a fluorescence microscope, it excites a specimen with a specific wavelength of light and forms an image of the fluorescent light emitted by the object. And what does it mean? First, you take the specimen, you coated it with a fluorochrome, and after coating it with a fluorochrome, you will expose the specimen or excite the specimen with a specific wavelength of light. And what is the exposing wavelength that you use? You can use either ultraviolet, violet, or a blue color light, depending upon what fluorochrome you are using. And as a result, what will happen? The A bright image of the object is being formed because from the fluorescent light which has been emitted by the specimen. The fluorochrome within the specimen will emit a corresponding light in response to the UV or the violet or the blue light which you exposed it to. And as a result, you will be able to get a bright image. And another feature which we have to say is, since the specimen is being illuminated from, since the illumination is being carried out from above the specimen. Okay, usually when we view anything under the microscope, we view things, the light source is being found below the specimen. But in the case of fluorescence microscopy, since the illumination is being done from above the specimen, it is also called as epifluorescence illumination or epi-illumination, or sometimes even called the reflected light illumination. And here, objective lens, which can also act as a condenser, is being used, and the specimen is illuminated from above rather than from below. So that is the reason why we call fluorescence microscopy also as a epifluorescence microscopy. Now let's move on to the parts of a fluorescence microscopy. It mainly includes different parts like a mercury vapor lamp, an exciter filter which transmits the desired wavelength of light, a dichromatic mirror which will direct the excitation light down the microscope, a mirror which reflects the light of shorter wavelength but allows a light of longer wavelength to pass through. So it will, shorter wavelength light will be reflected and the longer wavelength light will pass through it. And the excitation light continues down, passing through the objective lens to the specimen. You might wonder what are all these when I show you like this, but when you just look into the picture, it's more easy. So let's look into the picture right now. So basically, if this is your specimen, which has been coated with your fluorochrome, okay, and as I told you, you should have some light which will excite the fluorochromes which have been attached to the specimen. And that light here is being provided by the mercury arc lamp. Now, mercury arc lamp will produce some light and it will move forward. And what will happen? It will pass through an exciter filter. So, the exciter filter, what you will do is, it will remove all the long wavelength and only the shorter wavelength light is needed to be passed. So, it goes through this way. Okay, so you should remember only one beam of light or a shorter wavelength light is needed to be excite the fluorochrome, and it goes in knocks at the dichromatic mirror, and the dichromatic mirror will reflect the short wavelength of light and transmit. It will just so the shorter wavelength of light, it will come and fall onto the specimen. When the short wavelength of light comes and falls on the specimen, the fluorochromes which have been found to be attached onto the specimen will get excited. So it absorbs a shorter wavelength light radiation and it emits a longer wavelength light. Now this longer wavelength light is again passed through the dichromatic mirror. Then what happens? You also have a barrier filter over here which will block any ultraviolet radiation, but it will allow the visible light to pass through, and then you will be able to view your image over here. So this is the main principle of a epifluorescence microscopy. You are just basically exposing the specimen with a excitation light, and this excitation light is a short wavelength light, and in response to the short wavelength light which the specimen is getting exposed, it will emit a longer wavelength light, and this longer wavelength light is passed through a dichromatic mirror which permits it to move ahead, and there is a barrier filter just in case if any ultraviolet light or something like that is emitted from the specimen or from the system or something. To avoid that, you also have a barrier filter over here. So I guess you got an understanding of what is epifluorescence microscopy. I will just show you another depiction of the same thing. You have the mercury arc lamp here, which is emitting different lights, and I told you short light as well as long wavelength light will be emitted from the mercury arc lamp, but only the short wavelength of light is needed to excite the specimen. So you have an excitation filter which will block the what the longer wavelength light, and the shorter wavelength light only falls on the specimen, which absorbs the shorter wavelength light, and in response the sample emits a longer wavelength light which passes through the dichromatic mirror, and you also have a barrier filter or the emission filter, and finally it is being viewed by your eyes. So the fluorochrome absorbs light energy from the excitation light and it fluoresces brightly, and the emitted fluorescent light travels up through the objective lens into the microscope. And because the emitted fluorescence light has a longer wavelength, it passes through the dichromatic mirror to the barrier filter, which blocks out any residual excitation light. And finally, the emitted light passes through the barrier filter to the eyepiece where we view the specimen. Now, what are the uses of these fluorescence microscopes? Sometimes you can see that certain bacterial pathogens can be identified after staining with them with fluorochromes. Or sometimes specifically you can tag them with fluorescently labeled antibodies using immunofluorescence procedures. Now you might think, what is a fluorescent labeled antibody? An antibody is something which is being formed by your body against the antigen. So on the surface of the bacterial pathogen, there would be certain surface molecules, and they are considered to be as the antigens. And the antibodies which are being found to be specific to this bacterial antigens will be synthesized in the laboratory, and they will be coated with fluorescent molecules. So if your sample contains this pathogen, what happens, and you put the antibody will go and specifically bind to the antigen, and when you view it under the fluorescence microscope, what happens, the fluorescent labeled antibody will be fluorescing. And in the case of ecology, you can see various photosynthetic microbes as because their pigments, they naturally fluorescence when excited by light of specific wavelength. And sometimes even to distinguish between live and dead cells, we use fluorescence microscopy. Now, acridine orange can be used for live and the dead staining. You can use any fluorochrome, and also for the localization of specific proteins within the cells. For these purposes also we can use a fluorescence microscopy. So I guess you are clear with fluorescent microscopy. In this case, what is this? The first figure which we are showing here is a live cell which will fluorescence green, and the dead ones will fluorescence red. So the live and the dead, the distinguishing of the cells which are being living and dead is being possible by by exposing them with fluorescent dyes. And this is another case where the fluorescent antibodies tag specific molecules. Here the antibody which we are used in this figure, it is binding to a molecule which is unique to Streptococcus pyogenes, which is responsible for causing strep throat. Okay, so these are the two uses of fluorescent dyes as well as the tags. The first one, the live dead distinguishing, and the second one is a case where a pathogen is being detected using the fluorescent microscopy. So I guess you are clear with this session. Thank you for now. For any queries, you can just put your query in the comment box. I would be answering it depending upon my the knowledge which I can give you. Thank you for now.